of how many ATPs? 2 ATPs. And we end up with this molecule called pyruvate. Of course, this is the ionic form of pyruvic acid. This is pyruvate kinase, a pretty big enzyme, and I think this is also a major, important regulation point in this pathway. Yeah. So, the way that ATP has been produced so far in this glycolysis is by phosphorylation at the level of the substrates. Okay, so this is the substrate, for example, this one here, and it donates its phosphate group to ADP. This is catalyzed by the enzyme to make ATP. Yeah, question up here?
So because there are 2 of those going through glycolysis at any given point, does that mean that it leads to 2 pyruvates at the end?
That means also that there are 2 pyruvates at the end, exactly, for each glucose molecule. So here's the summary then. So we made an initial investment of 2 ATP molecules to break down glucose into 2 parts, 2 3-carbon sugar molecules that then become pyruvate in the end. This is an acid. But we get a payoff of 2 ATPs. Right? That's the profit. Also, we've reduced 2 molecules of NAD, now storing that energy in this molecule, NADH. So that's glycolysis. Simple, right? There's a lot more complexity to glycolysis. But let's talk about the citric acid cycle. So this citric acid cycle and the following step are occurring in the mitochondria. So somehow we need to get pyruvate into the mitochondria. Also, you may remember that mitochondria are evolutionary derivatives of bacteria. New bacteria. Exactly right. So that's why they have their own genomes that have a weird genetic code and so on.
Actually, that's just a theory still, isn't it? Is that ever conclusive?
Just a theory, but there's a lot of good evidence to support that. In fact, we maybe can cover that in the next course, molecular cell biology, or take my course in evolution. So one important aspect of these mitochondria are these membranes here. So there's an outer membrane and there's an inner membrane. The inner membrane is called cristae, and in mitochondria they're folded all over the place like this to increase the surface area and to maximize the amount of enzymes, the density of enzymes that occur in these sheets, in the membranes. I believe the enzymes in the Krebs cycle, in the citric acid cycle, actually occur in this area, in the matrix of the mitochondria. This matrix is really a dense gel, so probably all of these enzymes are really packed very closely together, so they can feed off of one another. The substrates are right there, and they can chain reactions together. So after you get pyruvate, you can go 2 different ways. One way is by fermentation. So if there's no oxygen present, like for example, you've been running, and running 2 miles, 3 miles, 5 miles, your muscles are getting really, really tired, and there's no oxygen left in your muscles. It's really not lying after 5 miles. Then your muscles can actually make lactate instead of going through this cycle. There's other organisms that can make ethanol, so this is obviously very important for some of us. Like yeasts, for example, make ethanol, so do some bacteria. There's important bacteria that also make lactate. Right? If you eat yogurt, then you know that this is very important. important step, because in the absence of this cycle here that we'll talk about, this gives you a way, it gives the cell a way to recycle NAD plus from NADH. So you've stored up all this electron energy from NADH, and if all of NAD is reduced, you don't have any NADs to accept more electrons upstream. So it's important to recycle that molecule. So this gives one way to recycle that. And if you're interested in more details in fermentation, read that. But we won't cover that in lecture. Okay, so how do you get pyruvate into the mitochondrion and get to the next step? So pyruvate is transported actively into the mitochondrion with this transport protein that's on the surface. And then there's this enormous complex. It's a huge protein complex of several different enzymes, and coenzymes, and things. That's even larger than the ribosome. It's called pyruvate dehydrogenase, and it catalyzes these three reactions. So the first thing is decarboxylation, so we can make CO2. We actually take off this CO2 here from pyruvate. So that's the first molecule of carbon dioxide that is made. So this thing is completely oxidized now, and it is just a waste product. Make as much energy as we possibly can out of that part of the molecule. The next step is that this oxidizing agent here, NAD, is reduced. So there's electrons that are then transferred to NADH. And this acetyl group is attached to a coenzyme called coenzyme A. So this coenzyme looks like this. What is this? It's what? A and P. Okay, that's a good start. So this is adenine again. This is a ribose. And it's a phosphate backbone there. Yeah, so that looks like a nucleotide, all right. What about this thing here? That's totally unfamiliar to me, for sure. It's a derivative of pantothenic acid. I don't know if you've ever been to the vitamin stores recently. So you see under P, pantothenic acids. This is essential for you, so you have to get it from outside. And that's what it's essential for making coenzyme A. And then there's another part over here. This has a sulfur group, so it's a mercato something. And this sulfhydryl here is what actually attaches to this acetyl group from pyruvate. So because of that sulfur, this and this is a very high energy bond. And that's the reason why you see this like little S shape. It just means that it's a high energy bond. So we have this 2-carbon derivative of the original glucose attached to acetyl-CoA, and this is the starting material for the citric acid cycle. So these are enzymes and substrates that will be in the matrix of the mitochondria. So acetyl-CoA has these 2 carbons here, and they are transferred to this starting substrate, oxaloacetate, to make a 1, 2, 3, 4, 5, 6 carbon acid structure, called citrate. So it's known as citric acid if you have the H minuses. There's a hydrolysis reaction here. This